Hello, everyone. Welcome to the CIT lecture for today. It is my great pleasure to welcome Giulia with us today. I'm very excited for the talk. So Giulia Baracchini, and correct me if I said your name wrong, is a PhD candidate in the integrated program of neuroscience at McGill University. She holds a BSc in psychobiology and an MSc in cognitive neuroscience and clinical neuropsychology from the University of Padua, Italy. She also trained at the Freie Universität Berlin and the Rothman Research Institute in Toronto. While her training was mostly clinically oriented, she became interested in fMRI and aging research while working with Dr. Cheryl Grady in Toronto. She's now pursuing her PhD in Dr. Nathan Sprang's lab. Her current research focuses on understanding how fMRI-derived pulse signal variability, a proxy of macroscale brain dynamics, contributes to the brain functional network organization in space and over time. So thanks again for coming, Giulia. So for everyone, just a reminder, please keep your microphone muted during the talk. Giulia is going to have 3 opportunities during the talk where she's going to pause for questions. So feel free to raise your hand at that time, and then I can designate you if you want to ask your question out loud. Otherwise, you're free to put it in the chat, and I'll be happy to ask it on your behalf. And otherwise, well, the floor is yours.
Thank you. Okay, yeah, thank you for inviting me, and I'm very excited today to present to you some of my PhD work linking to core aspects of spontaneous brain activity. As Fred said, one is both signal variability and functional connectivity. And before we go into the content of today's talk, I actually want to have everyone on the same level and describe what variability is and why it's important. If we think at a broader scale, variability is a change in some measurable quantity. And variability is actually present a lot in our everyday lives. And I was trying to come up with a relevant example that could drive home the point that variability matters. And I don't know if you were here 5 seconds ago, but Malar asked me if I recovered from my ankle injury, and I'm a runner. And if you run a lot, you know that running variability actually matters. And if you're well-versed in the running field, in the running world, you know that high-performing runners like Eliud Kipchoge swear by running variability. And by this, I mean varying running paces, varying running durations, changing your running shoes, your running surface, and so on and so forth. And the message that I want to drive home here is that all these factors actually improve and maximize your running performance and economy. Now, while I could spend 45 minutes talking about running variability, let's go into brain variability and why it matters. So in today's talk, I will argue that brain variability matters, and I will walk you over and trying to explain how we quantify variability via fMRI. I will then hit some factors that we should keep into consideration when we either are interpreting variability findings or conducting our own bold variability research. And then I will move on to 2 studies. One is a study that was published last year in your image. That is actually the title of the talk, so trying to find a link between variability and connectivity. And then I have some fresh results looking at these 2 aspects in healthy aging, and I'm still interpreting these, so bear with me on those. And then I will move to some future directions and conclusions. So why brain variability matters? Well, variability is present at every level of the central nervous system. And this is, from the sensory information that is incoming to the motor output that we're executing. And because variability is ubiquitous and pervasive, we should take this metric into account in our own analysis. And it will now be the case that, we should appreciate the ways in which variability is influencing both brain structure and function. And for the purpose of today's talk, I will tell you how variability influences brain function. Now, throughout the years, there have been a lot of theories that have been put forward to try and understand what variability actually underlies, what kind of processes it may underpin. So one idea is the idea of stochastic facilitation that in a way states that variability is this relevant noise that enables effective neuronal function. Another idea is that variability allows us to produce probabilistic responses. And by the virtue that we're producing probabilistic responses, then we're able to adapt to an environment that is changing. Another idea put forward by Randy McIntosh is that variability may be this kinetic force or moving force that allows neurons to explore states and gain access to the dynamic repertoire. And here I'm showing two scenarios. These colored in three different colors or three nodes. On the left, we have a scenario where we don't have variability. And then on the right, we have a scenario where we have variability, and this on the bottom are called manifolds. So in the absence of variability, what's happening is that the three nodes are not really exploring the different states or different configuration of the system. On the other hand, when we're adding variability, what's happening is that the same three nodes actually now are tracing various trajectories. So they're able to explore different states. So while, at first glance, these three theories may in a way be different or telling us different stories, they converge in trying to tell us that brain variability actually matters and it may be beneficial. Now, if we look at the fMRI literature on bold variability, there have been throughout the years, and this is work primarily led by Dag Garrett, now in Berlin, so all this work has shown how brain variability successfully predicts behavior. And this is within and across individuals, at rest and during different tasks, in healthy younger adults, in clinical populations, and so on and so forth. And what's really interesting is that greater variability, so greater bold variability, is associated with greater accuracy and faster reaction times. And so in summary, it's associated with better cognition. And so together, if we tie together all these concepts, what we can derive is that variability is an intrinsic characteristic of optimal neural functioning that allows us to predict and respond to the changing environment and efficiently process incoming information. Now, what do I really mean by bold variability, right? Like, how do we quantify this metric? If we take a step back for the people in the audience that are not that familiar with fMRI, what we are measuring using fMRI is called a bold signal. So here is an example. I have my brain, I have two brain regions, X and Y. And this is what the bold signal actually looks like. So what I'm measuring is the activity in these two brain regions. So on the x-axis, we have time, and then on the y-axis, we have a bold signal. Now, depending on the scanning protocol that we are implementing in our own study, if we are looking at spontaneous brain activity, what we may be interested in is in looking at how these two regions over time are correlated with each other. And this is called functional connectivity. If we have a task design, that what we may be interested in is in taking the mean over time of the signal in each brain region. Now, what I'm talking about when I say bold signal variability is a deviation over time in each regional bold signal. And if we think about statistics, an easy way to calculate variance or variability is by taking the standard deviation. Now, this is just one way we can quantify bold variability. Another way, for instance, it's called the mean square successive difference. And in short, and from here on, I will refer to this as MSSD. Now, one thing that I do want to highlight is that these two metrics are very highly correlated, and just keep this in mind for future slides. Now, how exactly do we compute MSSD? So again, we have our toy brain, this is region X, we have time on the x-axis, we have bold signal on the y-axis, and instead of using the standard deviation, so taking the overall change over time, what we're doing with MSSD, we're just calculating a series of differential equations or differences, where we're subtracting the single and one time point from the next, and then we keep repeating this process until we get to the end. So what we're looking at here is just a moment-to-moment change in the bold signal, right? And this is independent of shifts in the mean. Now, this was an example of the different ways we can calculate variability. And this kind of alludes to the fact that there's a lot of heterogeneity in the methodological side of bold variability. But the variability metric, so the fact that we can calculate variability in different ways, just one piece of this puzzle. There are a lot of factors that we need to consider, both when we are running our fMRI bold variability studies and when we're reading about these studies. And these are some of the factors that we need to keep in mind. So data acquisition protocols, so whether we're using single echo, multi-echo data, pre-processing pipelines, how we normalize the time series before we get to the variability metrics, or the metric that we're using, variability metric, and then if we're using task or rest. All these factors actually motivated me to write a review paper where not only I am summarizing or the state of the field in the bold variability literature, but also my goal is to give some advice and provide some guidelines to researchers that are either approaching the field of bold variability or trying to interpret findings of bold variability. And this is, motivated by my own personal experience, because when I first started getting interested, or when I was interested in this topic, I was kind of confused and I didn't really know what to make most of the times out of these results. And increasingly so in the context of, reproducible neuroscience, we want to be transparent in what kind of methods we're implementing to get to the results that we are now showing. So I find this work that I'm doing in collaborations with other colleagues that may be really important in the context of bold variability. Now, for the purpose of the present talk, I will only focus on variability metric. So as I mentioned a few slides ago, there's multiple ways we can derive variability: SD, MSSD, and then there's others. And I said that these two metrics specifically, so SD and MSSD, are very highly related. Now, this, if you think about it, may justify just using these two interchangeably. And while this may be okay, we need to pause for a second and actually think whether these two metrics, so SD and MSSD, are reflecting similar aspects of the time series. And if that's not the case, then we may have to interpret our findings accordingly. So I wanted to take a deeper dive into this topic, and I'm going to show you some time series simulations and real fMRI data that drives home this point. So if we just stop for a second and think about the formula of standard deviation and MSSD and how these two metrics are calculated, you can quickly see the standard deviation is more related to the amplitude. So it's the magnitude of change of the signal, right? And on the other hand, MSSD is just a slope, so the change or the frequency of the signal. So here first, I'm going to show you some regular sine wave simulations where I'm manipulating either one of these two aspects while holding the other constant, and then real fMRI data. So I'll start by showing when I manipulate amplitude and hold frequency constant. So I'm increasing amplitude, going from the top to the bottom. And as you can see here, both MSSD and standard deviation are increasing. What happens when I revert this scenario, when I manipulate frequency while holding amplitude constant? As you can see here, what's happening is that there is no change in standard deviation, in that the amplitude is actually invariant, right? I don't know if you can see, but the upper and lower limit of this regular sine wave is still between 2 and -2. So it's not surprising the standard deviation is exactly the same. But what's changing, and in this case decreasing, is the moment-to-moment change, so from one time point to the next. And this is reflected by a decrease in MSSD. And this is data, real fMRI data from two regions. So here again, you have time and bold signal, and I picked two regions that have contrasting MSSD and SD. So in green, what we're seeing here, it's a region that shows relatively slower frequency, so lower MSSD, but the amplitude is actually bigger, and so there's higher standard deviation. And then in orange, we have a signal where we have higher MSSD, so a more frequent moment-to-moment change, but a relatively lower or smaller amplitude, so a lower standard deviation. So what can we derive from these two sets of results together? Well, MSSD seems to be a frequency-dependent measure that is reflecting moment-to-moment speed of change. And on the other hand, standard deviation is more an amplitude-dependent measure that is reflecting magnitude of change. And because in all my future slides, so all my work is actually focusing on MSSD, what this implies for MSSD specifically is that this metric heavily depends on how and where in the time series you sample. So what is your frequency range and which time points are included? And this is even more so important in the context of task data. Well, the conclusion that I want to draw from this work is not that MSSD, you should pick MSSD over SD, or SD is at more advantages than the other. The point that I want to make is that you need to arbitrarily make a decision. So you need to pick either one metric to calculate your variability. But in light of these results, you should know what your metric is actually measuring, and then you should interpret your findings based on the metric used, and then test perhaps the generalizability of your finding using the other metric. So I just want to give you a brief summary of what I presented so far. We know that our brain is intrinsically variable, that variability is beneficial, not just for running, but also in the brain. And then using fMRI, you can estimate variability in different ways using standard deviation and MSSD. And then I hinted to some methodological considerations that you should keep in mind when you carry out and interpret fMRI-derived variability studies, such as variability metric. Yes, Gabriel.
Is this proper time for a question?
Yes.
I can wait otherwise. Yeah. Okay.
Yeah, it is.
So I guess in my work, I've been looking a lot at the measure of standard deviation. I'm working with mouse fMRI, so it might be a different story there. But I've looked at a lot of brain maps of the standard deviation distribution, but mostly for looking at confounds, actually. I found that by looking at a bunch of different data sets in mice, that it's a very good signature of motion or even physiological types of confounds. But that on the other hand, in subjects that have very little confounds, their distribution of variability tends to be mostly flat. So I guess when presenting this variability metric, I wonder, how do you distinguish between like a confound from a real bold part of the signal that might have a neural source? Was there some work that was done to validate how much you can really look at neural variability potentially?
That is a very important, and thank you for this question. Yes, so I know nothing about the mouse brain, so I can't speak to that, but I can speak to humans. Yes, there has been a lot of work trying to disentangle whether what we're looking at is related to, you mentioned motion, for instance, which is really relevant in the context of human neuroimaging. And what this work has shown is that it depends on your pre-processing pipeline. So you need to implement strict motion correction. And you need to clean your data before you get to even your bold variability calculations. And this is why one of the other factors that I mentioned earlier is the pre-processing pipeline, because depending on how good you are at cleaning your data, the effects that you are seeing may be either not present or actually be flipped in directionality. And there has been work looking at that, yes.
I'm wondering more specifically if there was, I don't know, maybe some electrophysiology or more ground truth measurements, because I've been, like in my work, I've been really focusing on exactly this aspect of the confound correction part of the literature, and I'm pretty well versed with the human literature on that. And as far as I know, I think there's really no way that guarantees that even with state-of-the-art correction, we have no leftover confounds that might, to some extent, still contribute to the signal. So I'm wondering if there's been some validation that we do indeed are measuring some neural part of the signal.
Yeah,
or is it related to brain networks? Maybe, or it doesn't have to be electrophysiology, but maybe that's where you're getting in later.
Yes, exactly. And one other thing that I do want to highlight is that if you, depending also on what type of data you have, so if you have multi-echo data, for instance, it has been, there's a lot of literature showing that, multi-echo is leveraging the biophysical properties of the bold signal. So I can separate bold from non-bold, and then I can calculate variability on bold. So they're having variability calculated on multi-echo data is actually a great argument to say that this is neuronal in origin, if that makes sense.
Yeah, I don't know the details of, I'm not that well versed in multi-echo, but I would be, from what I know about it, I would be more confident indeed that it's doing a better job at distinguishing the two. But all right, thank you.
Giulia, if that's okay with you, since we have two other questions right now, do you mind taking them?
Yeah, sure.
So first up, we have a question from Colin.
Hi, quick question. So also, I do share all Gabriel's concerns about the issue of motion of the compound variables, especially because I work in psychosis where it's a big problem in most mental health and also in aging. But anyway, I don't think there's any solution that's perfect cleaning your data. Just as an additional curiosity though, how do you feel about applying filters to the resting state data before variability? Do you normally filter, and what range of filters do you use? Because I feel like it would have potentially quite an impact on the MSSD in particular.
Yeah, that's a great question. So the ways I have calculated variability in my own work is actually using multi-echo data. So the filtering, smoothing, all of those steps are not really applied because I don't have to. But
people don't filter multi-echo data.
Sorry.
So it's not typical to apply filter to multi-echo data.
Yeah, I think we are just filtering the standard 0.1, 0.1,
yeah.
0.08, I think.
That's what I was wondering.
Yeah.
So are you guys running the 0.1 to 0.01 or whatever it is? I forget what frequencies we use now.
Sorry, I didn't hear the last bit.
Sorry, are you running the filters on your data before you do the MSSD?
Yes. So we're restricting it to the big, like 0.01.
Yeah, the standard.
0.08. Yeah. But to speak to your point, I didn't mention it here, but there is other ways you can calculate variability. As I don't know if you're familiar with FALF, ALF, and those are actually just picking, kind of, I think it's called low 4s, low 5. So there's specific parts of like frequency ranges. And yeah, that's going to be different, but I'm not familiar with that metric.
FALF's more of a power metric, right? It's the power in different frequency ranges. So it's a bit different than variability, but it's it's more amplitude dependent, I guess. Anyway.
Yes.
Okay.
Exactly.
Yeah.
All right, and we have another question from Lani. So she's asking in the chat, from your example, it looked like MSSD varied with both amplitude and frequency, whereas SD just varied with amplitude. So isn't MSSD more informative by capturing changes in both, or did I misunderstand the simulations?
Yeah. Hi, Lani. That's a great question. I don't know. I think like yes and no. I don't want to take sides because otherwise people are going to be angry at me. But I think so. And that's why I'm using MSSD. But yeah, the point that I want to make with these simulations is that depending on the metric you're using, you need to know whether it's impacting amplitude and frequency or just amplitude. So MSSD is impacted by both. Yes.
Right. Thank you, Giulia.
Okay. So now I would like to move on to my first study. So in this study, I am linking two sides of the same coin. So variability and connectivity. Oops, how do I... Sorry, one sec, I have this in the middle. Okay. So far we talked about the brain as a variable and dynamic system, but we know in the big area of network neuroscience that the brain is also interconnected, in that single brain regions are not working in isolation, but there is an orchestra of activity between them. Now, a natural question is, how are these concepts actually related to one another? And there has been extensive microscale work pointing to the direction that these two aspects are actually linked. And so part of this work has shown how neurons that belong to the same neuronal population also share or tend to form synaptic connections with one another, and neurons that share critical variability, and this is variability in the number of spikes or the frequency of spikes, also form synaptic connections with one another. Now, because I'm more well-versed in the macroscale literature, what does this actually mean in the context of fMRI? And when I was reviewing the literature, not just in fMRI, but also EEG, trying to see whether researchers have drawn links between variability and connectivity, I saw there was a lot of EEG work conducted by Branislav Misic here at McGill, actually showing that there is an association between the two. Now, in the context of fMRI, there has been a paper published in 2018 linking these two concepts. And in this example here, I hope you can see it, we have a brain region that is linked to 3 other brain regions. And the point that this toy example is actually making is that the more a brain region is speaking to other brain regions or forming functional connections with other brain regions, then the higher the variability in this single region or node A. And if we think about what functional connectivity actually underlies, so one idea is that it's underlying information transfer in the brain. And if we link this to the fact that from microscale work, variability is facilitating or may facilitate the formation of functional connection, then it actually makes sense that if a brain region is speaking to different brain regions, that it's not entrained to just like itself or one other brain region, but it's actually speaking, because it's speaking to more brain regions, that it's variable or more variable. Now, trying to, I pause here for a second because I was trying to get a better understanding of this link, and I wanted to try and investigate not just the magnitude of the association between connectivity and variability, but also trying to get to the details of how the topographical organization of this association may look like. Now, if we think about how connectivity is computed, connectivity, statistically speaking, is the correlation between two time series. And so it's an interregional phenomenon. Now, the way variability so far has been computed, it's at the level of single brain regions, so it's locally or intra-regionally. And if we want to trace a link between connectivity and variability that goes beyond just how strong this connection or this link is, then we may benefit from looking at variability also as an interregional phenomenon. And so if on the one hand, we have connectivity that is investigating the similarity in the activity between region pairs, then what I thought that it may be beneficial is to think as variability also as an interregional factor, where I'm looking at how the variability in region X, or how similar the variability in region X is to the variability in region Y. And this leads us to the near image paper that I talked about. And before I go on to the results, I do want to point out two methodological factors. So the first is that this study was conducted in healthy younger adults. And then speaking to what I was mentioning before, this is multi-echo data. So I'm not going to go into the details of this pipeline, but what I do want to highlight, and this in a way speaks to what I was telling to Gabriel before, is that thanks to multi-echo data, we're able to estimate both measures of connectivity and variability from bold-only signal. And so we're dramatically increasing not just the sensitivity of our measures, but also how robust these measures are. Now, taking a step back, how did I come up with an interregional connectivity matrix? Well, this is our brain again. We have two brain regions, and I calculate MSSD, so intra-regionally, locally in region X and region Y. And now, as I said, I wanted to look at how similar these two levels or these two variability levels are between regions. And now one way to do it is you can calculate the correlation, and this is what connectivity would be. Another way to do it, and this is how I did it, was to take the absolute difference in the MSSD in region X and the MSSD in region Y, and then I multiplied this by -1 so that higher levels of this metric that from here on I called IRS MSSD indicate a higher similarity in variability between region pairs. And this is pointing to the same direction as connectivity, where we know that higher values indicate stronger connectivity. Cool. So now we have connectivity and variability in the same space, right? So we have a region by region matrix where each entry here indicates the strength of the connectivity, and here the strength of the similarity in variability. Now, before I go on to my variability results from the near image paper, I do want to pause and show you how these two metrics may be related if we just pick regions based on their local MSSD values. So this is work that I did with my undergrad. Here, what you can see is a distribution of local or intra-regional MSSD values, and we split them into low, medium, high. And the idea that we had here, we wanted to select pairs of regions that either showed similar local MSSD values, so low, low, medium, medium, high, high, or also create other scenarios where the two regions were showing different patterns of variability, so low, medium, medium, high, and so on and so forth. What I'm showing here on the right is a distribution of interregional MSSD. And one thing that I want to point out, I don't know if you can see, is that low values, by virtue of the fact that I added 1, I don't know if you remember, low values here are from 0.65 to 0.75, and the maximal similarity that you can get is 1. Now, how do these different scenarios impact connectivity and interregional variability? On the top here, we have a scenario where we have two regions that show similar local intrinsic variability. And so this is low. And as you can see, both connectivity and interregional variability are really high. On the other hand, on the bottom, we have a scenario where the local variability in these two regions is dissociating. So we have low and high. And as you can see, the variability and the interregional, the connectivity and the interregional variability are both lower. So now, keeping this in mind, what did we do in the near image paper? Well, we asked the questions, do regions that show high connectivity strength also show similar variability patterns? And so what we did was we correlated these two metrics across people, and this is what the result looks like. In red, we have greater association, and then in blue, we have lower association. And then, as you can see, what's happening here, what I'm showing is that there is a correspondence between connectivity and variability, and it seems maximal along the diagonal. So these are within network connections. Now, if we look at this matrix, it really nicely recapitulates our connectivity matrix, right? And so I wanted to make sure that what I'm looking at was not entirely driven by the variance in connectivity. So I normalized these two metrics by separating within and between network connections, and I repeated this process at the network level. And so this is what I'm showing here. These network level results are actually recapitulating our region level results, in that for all within network connections, so here along the diagonal, I'm showing a positive association between connectivity and variability. But the other important factor that this network matrix was actually showing is that as we move down this matrix, and this would mean going from lower level regions such as visual and motor to higher level regions such as control and default, what's happening is that these regions, which are thought to be hubs in the brain, are not showing only a positive association between connectivity and variability within their own networks, but also across, so for between network connections. And this kind of makes sense if, again, we think about connectivity and variability as this information transfer process. So what I showed here is that interregional variability is a meaningful metric, and that interregional variability maps onto resting state connectivity. And so what we can conclude from this first study is that if variability and connectivity are codependent aspects of brain function, then integrating measures of interregional variability and connectivity in the same framework may help us better understand the brain's functional network organization. Now, if I can take questions if there are questions here, otherwise I'll move on to the second study.
For now, I don't see any in the chat, but yeah, I did. So I think we can maybe move on for now.
Okay, sounds good. Oops. Okay, so no.
Sorry, Giulia, I went too quick. Yes. I saw a hand raise. The hand got unraised, but I didn't see who it was.
Oh, hi,
yeah.
There we go.
Hey, Giulia. Do you want to comment on how bold variability and connectivity differ in like the clinical population? Because what we did was like healthy young and healthy old, but I guess variability would not map onto resting state connectivity in like patients with dementia, for example. Do you want to comment on that?
Yeah, great question. I will keep that question, if you're okay, for my second study, because in this study, I only commented on how they're associated, right? Connectivity and variability. But in aging, we're gonna see how they are dissociated, not just associated. So if you are okay, I can respond to that later. Is that okay?
Of course, yeah.
Okay. All right. So now I'm gonna move on to my second study, which, as I said, it's in Italian, we would say fuori dal forno, which means like it's very new. So bear with me, I'm still interpreting the results. But what motivated this the study in healthy aging? Well, we know that age is a significant contributor of inter-individual differences in both signal variability. And what has been shown consistently is that in older adults, there is a decrease in local, so intra-regional both signal variability. And this has been shown at rest in various tasks, whether there are motor or visual tasks, but also tasks such as an NBAC task, and throughout different blocks with different cognitive loads. Now, what's also interesting is that older adults are not only showing a decrease in bold variability, but this decrease in bold variability is associated with overall worse cognitive performance. And so if we think at my example of why variability matters, and this in a way speaking to the idea that variability is beneficial, because in older brains, so this is a population that is showing decreased bold variability, then we also see worse cognitive performance. So what may be happening is that aging may be exerting some sort of dynamic compression of bold variability. Now, given these alterations in local or intra-regional variability, I asked the question, can we observe alterations in inter-regional variability with age? Now we know that there has been a lot of work showing that there are age differences in functional connectivity. And this is work also coming from our own lab, showing how in older adults, our brains become more functionally integrated, right? So within network connections are decreasing and between network connections are increasing. Now, building on the Neuroimage paper, if we know that interregional bold signal variability traces functional connectivity, the question that we have here is, can we observe similar age-related differences also in interregional bold variability? So in this study, I used the same data set as the Neuroimage paper, and then I added older adults that are coming from the same sample. Again, it's multi-echo data. And because we have this aging component, I wanted to maximize the differences that we know in functional network organization in older brains. So we took an individualized parcellation approach to define parcel boundaries and parcellate the brain. The primary metrics of interest here are connectivity, local and interregional variability via MSSD. And we also have behavior, given that we saw that there is an association between decreased variability in aging and worse behavior. And in terms of behavior, I wanted to look at both externally driven or neuropsychological or test-driven behavior, such as cognition, these are metrics such as the episodic and semantic memory, executive function, processing speed. I also wanted to look at internal cognition. And so these are metrics that are derived from the autobiographical interview. And we also had NIH measures of emotion. And here I averaged positive and negative emotion. So I'll first show you, nothing surprising, which are the connectivity results. So just to guide you, here on the left, we have our younger adults. On the right, we have our older adults. In red, we have pairs of regions that are showing higher connectivity. And in blue, we have pairs of regions that are showing lower connectivity. And as you can see, older brains are recapitulating what we know about aging. So the fact that there is an increase in red. So there's more between network connections. And this is also showed in our aging paper from our lab. Now, how does variability look like in the sample? So first, what I wanted to do is calculate local variability and test whether in our sample as well, we could replicate the finding of a decrease of variability, of local variability with age, or in this case, in older adults. So this is how I did it again, just to reiterate, I took for every region the MSSD score. And these are our results. So what I'm showing here are regions that are showing an increase in variability in younger brains. Or if we flip this around, these are the regions where older adults are showing a decrease in MSSD. And we don't find any regions where the opposite or the reverse is true. So together, what we can say is that older brains are expressing lower local MSSD in line with all the findings that have been already are present in the literature. And this is increasingly so in motor and visual variants. Okay, so nothing new so far. What about our interregional MSSD metric? So this is again how I derived our interregional variability metric. And now I'm going to show you what's happening. So on the left, we have our younger adult interregional MSSD metric. On the right, we have our older adult interregional MSSD metric. darker red colors indicate lower similarity in variability between region pairs. And in higher, lighter red, we have a higher similarity in variability between region pairs. And as you can see, just at a first glance, it looks like older adults are showing lower similarity between in their variability levels between region pairs. So keep this in mind as we move forward. Now, what I wanted to do based on the Neuroimage paper, I wanted to associate these matrices of interregional MSSD with connectivity. And so I correlated the two. Here we have younger adults, older adults, and I do want to highlight, speaking to what Igor was saying, this color right here. So in red, we have regions where there is an association between connectivity and variability. And what this means is that these two metrics are either both going up or both going down. Independence means that these two regions are not associated at all. And dissociation means that connectivity is perhaps going up and RSMSD going down, or vice versa. And what we can see is that in older brains, we're not just seeing more red, so more association between connectivity and variability, but also more blue, so more dissociation between connectivity and variability. It may be easier to actually visualize what I just said here in our network level matrices. And here I highlighted both lower level and higher order networks where both patterns of association and dissociation actually come out. This is the default, for instance. Here we have ventral attention, dorsal attention, sorry, and then we have visual and somewhat motor. Now, these are all great results, but are just descriptive, right? So I wanted to take this a step further and actually quantify these differences. Before I go into those results, I just want to summarize what we saw so far. So older brains have lower within network resting state connectivity and greater between network resting state connectivity. They have lower local MSSD, so local variability, and also lower interregional variability, specifically between network. When we add age to the mix, what this means is that it's important to investigate not just where connectivity and variability are associated, but also where they may be dissociated. And it's also what age is highlighting is that there is an interesting dissociation between within and between network connections and lower and higher order networks. Now, because I was interested in kind of disentangling and getting a better understanding of this association and dissociation between connectivity and interregional variability, I looked at them separately. So first, I'm going to present findings about the association or the age differences in the association between connectivity and variability. So this is how I did it. We have our younger adults, our older adults, our association matrices. I fed them into partial least squares, which is a multivariate technique. And I asked the question, which are the connections that maximally explain the age difference in the association between connectivity and variability? And these are our results. So what we can see in red, these are the connections that are showing a stronger association in connectivity and variability in older brains. And as you can see, these are between network connections, specifically for lower level networks, so visual and somewhat motor. In blue, we have those connections that are showing a strong association between connectivity and variability in younger brains. And these, as you can see, are primarily within network connections, specifically in heteromodal higher order networks, such as the ventral attention and the default. Now, what I find that is very interesting is that when you relate these brain patterns to behavior, this is what you find. So again, I want to orient you to this plot. Here are our younger adults, and here we have our older adults. In the different shades of pink, we have our cognitive or externally driven cognitive variables. In the different shades of orange, we have our internal cognition, and then we have emotion in green and blue. So while cognition is not significant because the confidence intervals are crossing zero in either our younger or older sample, what's happening is that internal cognition and emotion are both loading onto the brain patterns that we just saw, but the directionality of this effect is actually flipped. So for our brain results, what we saw is that there is a dissociation in where the relationship between connectivity and variability is maximally expressed within network connections for younger adults and between network connections for older adults. And then in terms of behavior, what we saw is that there is a dissociation in how these relationship patterns relate to internal cognition and emotion in younger and older brains. Now, if we look at, so this was interesting, but I find that the most interesting part is to look at, where connectivity and interregional variability may be at odds, because that in a way speaks against what we know about what variability may be underlying. Now, how do I do this? So we have, as I said, two groups, right? We have our younger adults, we have our older adults. And what I did was I separated connectivity and variability and I fed them into PLS, so partially square, as two separate conditions. And by doing this, I could quantify both the main effect of condition and also an age by condition interaction. So the question that I wanted to ask here was which connections maximally explain the age difference in the dissociation between connectivity and interregional variability, and how are these related to behavior? Now, to no one's surprise, I'm still trying to wrap my head around these brain results, so I won't show you the brain results because I'm in the process of interpreting them. But what I do want to show you is the behavioral pattern, because I find them really cool. So here on the top, we have our connectivity. Here on the bottom, we have interregional variability. On the left, we have our younger adults, and then on the right, we have our older adults. So at first glance, what we can see is that younger adults are not really telling us anything, right? So nothing is significant, either for connectivity or interregional variability. But if we look at our older adults only, what's coming out is that connectivity and variability seem to be explaining behavior differently in older adults. And if we look at them more specifically, connectivity is actually describing or is relating to the dissociation, so the brain patterns that I didn't show you, in terms of cognition, both external cognition and internal cognition. And this is not the case for interregional MSSD, but for emotion, then both connectivity and interregional variability are showing this pattern. So I'm still working on the interpretation of these findings, but perhaps what this may tell us is that maybe interregional variability is more a trait-based metric instead of, a trait end state, which is what connectivity is actually highlighting. So in summary, in this study, I showed that age is a significant contributor of interregional differences in both local and interregional signal variability. I also showed that age uncovers both association and dissociation patterns between connectivity and variability that separate within and between network connections and lower and higher order networks. And I also showed how age differently impacts the relationship between connectivity and cognition and emotion, and variability and cognition and emotion. So my takeaway here is that to better delineate how age influences the brain's functional network organization, we may benefit from looking at variability as an interregional phenomenon. And so there may be more than just connectivity alone or local variability alone. And this, integrating also interregional variability in our work, may tell us more about aging. Now, I do want to wrap this up and take us back to how I started this talk. And I hope I convinced you that interregional variability is a meaningful metric. So we know that our brain is variable and it's dynamic, and it's important to study local intrinsic variability as it predicts behavior. It's associated with age, but we also know that our brain is interconnected and there may be a link between variability and connectivity. And this is why I came up with a measure of interregional variability. Now, why is this metric important? Well, we know that brain organization has to be understood in an interregional fashion, and connectivity has informed our understanding of the brain's functional network organization, but this may be just one facet. And there may be aspects that could be uniquely explained by variability as also an interregional phenomenon. And an idea is this association versus dissociation hypothesis. So I'm not advocating that interregional variability is telling us the full story, but rather that perhaps this association and dissociation between connectivity and variability may be important. And this also speaks to Igor's question, because I am suspecting if we see this dissociation in aging, there may well be the case even in clinical populations. Now, before I move on to my last slide, I do want to give you a little preview of where I'm taking this next. So this is my OHBM poster. So if you're coming to HBM, please come to my poster. I'll be happy to guide you through my work here. But in summary, what I'm trying to do here is I'm looking at how stable connectivity and interregional variability are across the time series. So we are all familiar with dynamic connectivity, right? But the way variability has been computed so far is an average across the whole time series. But because the brain is dynamic, what if we do a meta? So we do variability of variability, and we look at how variability changes across the whole time series. And the idea is that in aging, we know that the aging brain is more stable or less flexible. So what I'm suspecting is that there is, these different windows of connectivity and interregional variability may be more similar. And there may be something also to say about these association and dissociation patterns when we look at these two patterns dynamically. And the way I'm doing this is by using co-status, which is a multi-table method of progression analysis. Again, happy to talk about this, but then I'm relating these both to age and cognition and emotion. And with that, I would like to thank you all for your attention, thank my supervisor, my lab members, and all our collaborators, and I'm happy to take questions.
Thank you, Giulia, for your amazing talk. For me, it really drove the point of interregional variability, how important it may be. So in terms of question, feel free to post them in the chat or raise your hand. And correct me if I'm wrong, but Giulia, I think you agreed for a student discussion afterwards if people had questions. So if anyone have questions but don't want to put it in the meeting right now, you can stick around after the presentation to ask your questions to Giulia and discuss. So first up, we have a question from Ranjani. He says, hi, I was wondering if the bold signal variability will be different in children as compared to adults. If yes, how different do we expect them to be?
Oh yeah, great point. Okay, so stay tuned, because I am now looking also at the whole lifespan. So I will have more answers to that question, but what I'm suspecting in some of the work that has looked at variability in children has shown that there is greater variability in children, lower variability in children. So there may be, I think it's an inverse U-shape. Yes, we call it an inverse U-shape, where younger adults are showing this maximal and optimal level of variability. But then as we're developing or getting older, then we have a decreased variability. But this is all work that needs to be validated and replicated. Yeah.
For sure, that's going to be interesting. Thanks for your answer. Next up, we have Gabriel with a question.
Yeah, sure. I was wondering actually a simple question. I don't know if it's common sometimes to look at how the brain variability map look like. Like, do you look at your subject brain maps of variability at each voxel and what kind of anatomical features it shows? Because in my experience, it's been quite useful to distinguish the quality of the metric.
Mm-hmm. Yes. So I never did variability at the voxel level. From a statistical point of view or standpoint, I prefer looking at regions. But that's a good question. And a lot of prior work, so the first studies that have been published from 2010, I think, on, they have looked at variability at the voxel level. So I can point you to those because I don't know the answer to that question.
All right, because my issue would be that when we already parcellate the brain a priori, then we've imposed a structure like the analytical structure we want to see already, but we don't know necessarily the source that's driving our measures when we average the voxel within the brain region.
Totally.
So if you're not going to be able to distinguish if there's residual confounds despite your multi-echo processing, for instance, or not.
Mm-hmm. Yeah,
so
totally. Yeah,
But
no.
yeah, if you can send over those paper, I'd be interested to
Yeah.
take a look.
Okay, will do.
Thank you.
Right. Next up, we have a question from Colin.
Hi again, great talk. Well, let me be the first to say clap, 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 because I feel everybody misses the audio feedback from, used to talking. You get your little. Anyway,
Yeah.
really interesting stuff. First, as a lifelong fMRI researcher, I have frequent, and this kind of goes a bit to what Gabe said, but always my question is emotion. I know you're working in multi-echo data, which might help that, but have you actually done any comparison to see if any of the associations you're seeing in regions are correlated with how much people moved in the scanner?
Yes, I have.
Good.
And they're not correlated.
That's really important, because if I got that as a review, is the first thing I would ask you, and I'd strongly recommend that goes in your supplemental material. Also, speaking a bit to what Gabriel was mentioning too, the idea of I'm totally on board with parceling the brain according to a reasonably well-defined parcellation structure based on functional data, but it might be interesting to think about which regions have more consistent internal variability amongst themselves. So there may be some ROIs that share a more, again, it goes back to the doing of voxel-wise, and maybe it's very esoteric and not entirely related to your question, but some regions might have more consistent, nearby voxel variability, and some regions might have a broader range of variability within a given region that might in some way be informative as to the local structure of how different regions interrelate to each other. Of course, there's always a million things we can ask and questions we can look at, and so you can do so much in the paper, but it immediately struck me as interesting. There was another thing too that was really, I know. This is a bit more of a devil's advocate question. Since connectivity is defined by the correlation between time series, wouldn't you almost automatically assume the variability would have to be high between connected regions?
Yeah, so that was actually a question that we got in our reviews. The way we did, yes, like I don't have an answer, like a full answer to this question. And I'm still methodologically trying to wrap my head around that. But one way we can do that is, correct me if I'm wrong, I can just look at the distribution of my connectivity values and then try and see the variability at the tail ends of this distribution, and then see what's happening. So stay tuned. I don't know.
Right. I mean, it's an inherently circular comparison in some degree.
Exactly.
You could argue also that it doesn't have to be true. Right? You can have high variability, high connectivity without necessarily high consistent variability or similar variability across regions, because you can get like low frequency components contributing to connectivity, but then one region having a higher frequency component on top of it, which changes the your variability metric. But anyway,
yes,
I feel like but there's an inherent circularity built in there, so maybe the relationship isn't surprising. I guess it's not necessarily guaranteed to exist though.
Mm-hmm. Exactly.
Right.
Yeah.
Sorry, that's for all the random things that popped into my head.
Yeah.
Great work.
No,
really interesting.
great.
If I do make it to HBM, I'll definitely try to hunt you down.
Thank you.
I'll jump in with a question of my own. So I'm as always, I'm super interested by the variability between individuals of the same group. So I'm just wondering, in your group of young adult, or in the literature, if you saw anything about like how both signal variability really varies from an individual to the next, or is it relatively similar within a group of individuals that are homogeneous in quotation mark?
Mm-hmm. You mean if I have different scans of the same subject, or within the same group?
Within the same group, if the variability changes a lot, like if your MSSD is quite variable between individuals.
Mm-hmm. That's a great question. Why would that be the case? I mean, you mean based on performance, for instance, or just looking at variability alone?
Just variability alone, because for instance, I'm working on fingerprinting. So like looking at how like functional connectivity profiles are almost unique from an individual to the next. So I'm wondering if that could be also reflected in the variability of the bold signal per se.
I think it could. I never thought it that way, but I think it would be an interesting venue of research. Definitely. Great question.
Thank you. And next we have a question from Colin.
Sorry, that totally made me think, because this is all I care about, is how similar or different people are right now. It's where all of our research goes. And I would be surprised if the variability was always super consistent across participants. What we've been finding in some of our work is when people who deviate from the more common patterns of at least during task activation, people who deviate from more common patterns tend to be the ones who show impairments in cognition. Now that's because that's during task fMRI, right? So it's kind of like the more typical and average your connectivity pattern is, or your activity pattern is. I mean, the more task engaged you are, and the better your cognitive performance tends to be, and things like that. You might find the same thing true in your variability metrics. So if you look at the older people, the people who show a spatial distribution of variability that differs the most from sort of the average or from the younger group might be the ones who are showing the greater cognitive impairments. And sort of the distribution of variability might become a metric of general brain function in some way. What we've been doing to look at this is just actually look at a distance between people. So we do correlational distance, and we calculate how far a participant is from other participants. You just average that, you can sort of see a typical there. Yeah. And it gives a very simple summary measure of someone's sort of typicality, which at least in our work in mental health has proven to be somewhat interesting.
Thank you so much for that, because it's actually what my COPE status measure. Whoops, where did it go? So this is a distance-based metric. Right. So what I'm looking at is so this is dynamically, right? So, but the idea is here, how far each subject is from the mean, if that makes sense. And this is what you just said, right? So I could actually look at this in this work, and that would be great.
I think the idea of typicality sometimes has a lot of benefit to it. There's kind of a generally optimized brain function pattern. And people deviate from that largely because I suspect of what we might loosely call damage. If some systems break down, other systems have to help compensate, which is why we see this change in pattern in aging potentially. So this sort of measure of like how much do you fall along the typical range is a sort of a surrogate for how well functioning is your network as a whole, and how much does it have to make up new ways to do the same task and probably using less efficient systems.
Yeah, exactly. I'm with you.
I love the dynamic stuff, by the way. This is actually what you were talking about. I was thinking it would be great to look at. So,
great. Great.
I'll shut up now.
So thank you for the great discussion. I see more questions in the chat, but unfortunately, we've reached the end of the time for the CIC lecture. So for the people who asked a question in the chat, I invite you to stick around for the student discussion so that Giulia can answer your questions directly. And thanks again, Giulia, for coming to the CIC lecture. That was super interesting.
Thank you. Thank you for having me.